Okay, so hello again to people that just joined. My name is Gloria Isabel Giraldo Calderon, and I'm an outreach officer for the Merge ViewPath database. Today, I'm also joined by other outreach officers, Omar Harb at the University of Pennsylvania and Sam Run at the University of Notre Dame. Behind the scenes, we also have other outreach team members that will help us answer all your questions. So there's a go-to webinar panel, and in that, there's a chat box, and there you can ask all the questions along. We are really excited to have you all here. This is the brand new website of VectorBase, the first time we show our merge resource to many of you, and there's been a lot of work behind the scenes. So we started back in September officially, but before that there were a lot of months of very hard work, and we are very excited to show you what that all means. And one of the benefits is that we have many new tools, ways to query the data, and that you have a much larger outreach team to give you support with all the needs that you have. Okay, so now let me switch actually to VectorBase. Oh, one last thing before I forget. This webinar is being recorded and will be available for you to watch again if needed. Same as with all the previous webinars. So let me go to VectorBase now. Can you see the VectorBase page, Omar?
Yes, I can see it. Looks good.
Excellent. Thank you. Okay, so once this webinar is finished, you can click here under Help, learn how to use the ViewPath resources, and here under Webinars, you have all the previous events that we have run, and for each one of them, there is a link. For example, this one, the ones from last week, and here is the recording, and also to the upcoming ones to register. So, for example, this one and the registration link. So don't worry if we go over too fast or you want more details, we have dedicated webinars and you can come back to this page. Okay, so first of all, I'm going to show you the layout of the website. So all the sites in ViewPath, which you can access from this bar here at the bottom, have the same interface, and that allows you to move from one to the other very easily. So today, of course, I'm going to show you VectorBase, but for example, if you are working in Anopheles and you are interested in anything about Plasmodium, it will be very easy for you to to move to that new website because it looks exactly the same, but now you are in Plasmodium and not VectorBase. Okay, so at the top we have here the name, and if you click on the logo, you can come back to the home page. We have the site search, which I will demo you how you can use it. And below we have these 8 navigation menus. So we have My Strategies and Searches and Tools. What you can do with them? You can not only download the data, but the power of the ViewPath resources is that you can query the data. This is a big difference from other repositories. You have also My Workspace, and here I would like to highlight Galaxy, in which you can upload your own data and do differential expression analysis, or you can do a SNP calling, and you can send or import the data back again to VectorBase with the ViewPath resources and continue with the search strategies. And you will learn a little bit more about what that means. We have the data, the about, help, which I already showed you, this very important one here, learn how to use the resources, and contact us. This one is very important for us. Please email us anytime. We're always happy to hear questions from both novice and experienced users. And we do all this for you. So please let us know things that you like about VectorBase and things that could be improved. Here on the side, we have the social media. So we have Twitter, Facebook, and YouTube, and also a link to your account, which is not necessary to use the website. but provides you with extra functionality, such as being able to save strategies that you construct to, for example, test a hypothesis. So with the site, you can run in silico experiments with in vivo data. And if you have an account, you can save that and share with colleagues or have it for later. Okay. You can actually close these banners if you want, and here we post important information for you. Let's expand this a little bit. Okay, on this left-hand side panel, we have search for, and this is used to build strategies. And I'm going to demo you, once I finish the layout of the homepage, I'm going to show you how to construct a strategy. The central part and more prominent area of the website is occupied by the overview of resources and tools. And these, we call these cards. And when you click on them, these are like very short and sweet guides to quick start how to use the website. So they are designed to be very brief, and they will show you just quick read more, 1, 2, 3 steps. And of course, if you want more detailed information, you can come here to the bottom. These are PDF files with workthrough examples and exercises step by step. And remember, you can also access them from help, learn how to use the view path. Okay. On the right side here of the website, we have the news and tweets. You click here to open them. The most recent of which is that we had a release in past May, and in this one, it will tell you what happened with the website. Well, of course, in this release was the new website per se, but we also normally will tell you which new data sets you got. For example, in the last one, we only got new data for the map view, sorry, previously not the PubBio map, but for the upcoming releases, we will have also new omics data sets. But that's all I want to say about the homepage layout. And now I will switch it to Omar, which will tell you about data sets.
Thanks, Gloria. And I'm happy to do this. You can stay on your screen if you want. It'll make it easier. So we want to do screen transfers. Okay. And just to highlight a couple of things. So the process of integrating both UPathDB and VectorBase obviously has taken a few months. We had to rebuild the infrastructure to accommodate both resources. But also we had to transfer all the data, both from VectorBase, and we're actually still working on transferring all the data through new pipelines and so forth in the new infrastructure for the Parasite databases. And this is an ongoing process. The databases themselves are available, as Gloria mentioned. And we did for VectorBase, we've transferred almost all the data from the legacy site, from the old site. All of the RNA sequence data, all the microarray data. There may be a couple of data sets that require additional work, and those are being worked on to transfer them. We're working on proteomics data, and one thing that I know Gloria stressed, and hopefully it will become very evident when she shows you the strategy system, is that the goal of resources like VectorBase is not just to display or allow you to display and look at a single data set at a time, but rather query the data. So allow you to ask your own questions and then integrate the data together with other data sets. So for example, being able to say, find me all genes that are upregulated in a delta methrin resistant strain or mosquito that also have proteomics evidence and are predicted to be secreted, right? So those are types of questions that you can integrate and work together. So if you click on the About menu, and Gloria, maybe you can demo that, and you will see that there's a section called Submit Data. And we often get a question, well, where do you get all your data from? How do you hear about it? And if you click on the How to Submit Data to Us option, there'll be a lot of information here, right? So there's information about what is the process, and about all the types of data that you can submit. So there's a lot of information here. You're welcome to obviously read all of this and go through it and find the type of data that you want to submit to us. Sometimes the easiest thing to do is just to click on the Contact Us link and say, hey, I have you know, a new RNA sequence data set that I'd like to make available to the community. Here's why it's important. And that obviously then puts it on our list of data sets to track. And then we can put them in the loading process at some point. We also work with the community very closely in scientific meetings and open community calls, which hopefully you will get an email soon about an open community call for VectorBase, and actually for different sub-organisms in VectorBase. And the goal of those is to really find out from you, the users of this database, what data sets are high priority, which ones, you know, should go in first. Are there any tools that we need to develop for you, and so forth. If you click on the About menu again, Gloria, and go to the Submit Data, there's another option there called Data Submission and Release Policy. This is just a PDF, but it's an extensive release policy. And really the bottom line of this document here is to let you know that we never release data prematurely. So we actually hear about lots of data sets pre-publication. And in fact, people send us data sets pre-publication because they would like the data sets to come out, you know, very close to the publication time, or sometimes, often actually more and more increasingly so, pre-publication as people put their data on bioarchives. And the goal of the document to say is that, you know, even if you tell us about the data or send us the data, we're not just going to load it and release it. We will make sure to communicate with you throughout the process. And once we have it on a private database, we allow you then to QA it and let us know, oh yeah, this looks good, here are some issues or no issues, and then give us the final approval to release. Bottom line is that you know, the goal is for you to be happy with the way the data is represented in a database, and then the database will provide you with all the tools and provide the community with the tools to be able to ask questions about the underlying data. All right, Gloria, I think this is all I was going to say about data submission. Feel free to add anything else, and but you can carry on, I guess.
Yeah, I think you said it all, Omar. Thank you very much.
Cool.
So now I'm going to show you how site search works. So site search is this box here, and this tool is familiar for the VectorBase community users, but new to all other ViewPath users. It's available not only from the homepage, but also from any other page. So to become familiar with the tool, you can search all the data that we have indexed there. So you can you know, have the wildcard there and just run your search. Okay, and notice here that we have some categories: genome, organism, transcriptomics, population biology, metabolism, data access, institutional, and about. And each one of those have subcategories. So if you filter down, you will get more results, and you can filter by organism. But this is just for you to get to know what is all the breadth of data that you can find if you use site search. But for example, let's say you are going to use a keyword. So let's use kinase and type search. So notice now I don't have all the other filters that I showed you before. Why? Because I have this here. So if I uncheck this box, so notice how I don't have hits for any of the other categories available. So that's why this is by default checked for you. Okay, so right now I have more than 40,000 hits in this page for this query, but here on this page I'm only looking at 20, and you can navigate through these different pages or with the arrowheads, but that's still, you know, a lot of them. So what you may want to do is to filter them. So I said, well, I only want to look at genes, for example. And I only want to look at for this in the snail, an intermediate host, Bioflaria. So I check this box and click apply. So from 40,000 hits, we are now to more than 1,000 hits. And say, well, I only want to see the hits that have a product description, and you apply that. And now we go to 82 hits. So instead of just going one by one, you can export this as a search strategy. Oh, sorry, before I go there, I almost forgot. So the way this works is this is a text search. So my word was kinase, And where did it find it? So if I open here, the fields that match my query are these ones, the Go terms, the orthologs, and here you have the word highlighted. Okay. Now notice that before, now I'm going to clear this filter. Notice that this is gray out and is not working. Why? Because I need to select a subcategory. And once I select a subcategory, this becomes active. And now I can export as a search strategy to download and data mine. So now I can click here. So I want my genes with my product description, apply, and now I export. Okay, and here are my 82 genes, and that query that I did became a single step. So now that was site search, and you can connect with the search strategies. And now what is the search strategy? It's a tool for data mining. Okay, so how I'm going to do this? You can, if you're just getting started, I will recommend also that the same way that with site search, I told you, well, just type the wildcard or the asterisk to get to know it. It's also good just to browse the categories. So I'm going to pause here a little bit and show you what I mean. So if I go back here, so searches from here are this same one, search for. And you can expand them all and notice that each one has other categories below. But you don't need to memorize them to use them. What I'm telling you is just to open them to become familiar and to know how to use them. Okay, so you take a look of the categories. You know what is there. You know what are the subcategories. But actually to use them, just type a word of what the thing, something that you are interested. So for example, you're interested in RNA-seq and you selected here. Where do you arrive? You arrive to a page with all the experiments that we have for RNA-seq data. And notice they all say new. So for the current VectorBase users, these are actually not new experiments. These are all the ones that come from our legacy site, but they're just new in this new website. What is actually new for you is the ability to query this data. Okay, so for example, let's say that I am interested in insecticide effects in Aedes aegypti larvae. So I'm not going to scroll to all of this. I'm just going to type a keyword, insecticide, insecticide, and I can already see it. So because I was look at here the organism. So I'm interested in Aedes aegypti, so these are the ones I want to look at, and I actually want larvae. So it seems like my data set of interest is David et al. And if I want to learn more about the data set, I just click here in the question mark, I get a nice summary and a link to the publication. You may have noticed that I have here these icons. If you hover with the mouse, it will give you a description of each one to use. The one that I'm going to select is this one, differential expression. Why? Because this one means that the experiment had enough samples for us to run a pipeline that will give you a statistical significance and p-values. If we don't have enough samples and we cannot give you statistical significance, you can use this fold change option, which is still good for exploratory purposes. Okay, so I'm going to select differential expression. And notice I have it here. Here's my experiment. What I'm going to do here is to select my reference sample. So I'm going to have larvae untreated control, and I'm going to compare this, my comparator sample, against the larvae that were exposed to permethrin and the direction of the differential expression. I only want the genes that are upregulated, and I'm going to leave this as defaults. The fold change to be 2 and the p-adjusted value, and I click on Get Answer. Okay, so notice this was my previous query, and now I'm doing another query, and here's what I get, my results. Okay, so see how I hover, when I hover with the mouse, I can see this edit option here. So if I click on edit, I get this panel and I can double check that actually I selected control, parametering, I've regulated. Yes, that is what I wanted to do. If I made a mistake, I just click on revise and I can, repeat the steps that I was going through. Okay, so we obtained 110 genes, and these are the genes I have here, and you have them here in the table below. What do I have in this table? So I have the gene ID and the transcript IDs. I have, I can add columns or delete columns. So for example, I want to add a column and I want to have the organism. The organism, for example, but in this case, it doesn't make much sense because this is an experiment just with SGPT, but I was just to show you that you can add or delete the columns. You have the product description here, and the last two columns, you can sort them, the value, so you can say, for the highest to the lowest p-value. One really nice feature here is that you have this link that in the product description allows you to see those genes, where is their product coming from. So I can see that most of them are actually unspecified, are pure proteins. So what can I do to know what is the function of those 110 genes? So that is a very good example of how to expand your strategy. So now this is step one. I'm going to add another step. And I'm going to transform my records. And I want to know, for example, if I add orthologs, because I know Anopheles gambia probably has, it's a better curated genome. One step, selected there. And now I have them. So I have the orthologs now. And notice how the number of genes changes. And now that this is yellow highlighted, so if I click here, I have 110 genes in this table, and these are Aedes aegypti genes. But if I click in the box again, I got 180 genes, and these are the Anopheles genes. So when you select in the different steps, you are looking at different results. Okay, now I want to click on the product description again, and I want to see what this data is about. And I see, not surprisingly enough, that many of them are cytochrome P450 genes, which are extensively studied when talking about insecticide resistance. So that's very good. So now I know more about these genes. And I can add a third step to my strategy. So for example, I can transform the genes again, and I want to know in which metabolic pathway they are. I'm going to run all these with the default options, but the pathway source is KEGG or MetaSeq. Pathways must contain any of the specified genes, and run step with the defaults. And now, notice that I have 216 pathways. Why? Because some genes can be part of more than one pathway. Okay. So if you are satisfied with your results, you can actually download them. And here's the link to do that. But keep in mind that you are going to download what you're looking at. So for example, I'm looking at pathways. This is what I'm going to download. If I click on the orthologs in Anopheles gambia, so when I click, that's what I'm going to download. So you need to, if you want to download every single step of your strategy, you need to do it one by one. Okay, just to finalize before we move to another section, let's go to public. Here's public. Okay, so Omar and I had made for you available 4 different strategies for your reference. You can click on the title of them to import them to your session. So you click here, for example. And now we have imported the strategy that Omar made for you guys. And when you import that, you need to save it and you give it a name. But this is very important, and it's in red here. So you're saving the configuration, you're saving the steps, the strategy, but not the data, because with every release, data may have changed, or we may have changed the pipeline to obtain the differential expressed genes. So the data will change, but your strategy will remain. And here with edit details, you can change the title of it, the description, and tell, oh, what I did in the first step, what I did in the second step, what I did in the third step, because today you may remember, but later you don't know why you did what you did. Not the strategies can remain private, but just in case you want to make them public, I'm not with that one. Maybe with this one we just run edit details. If you save, oh, sorry, I cannot find it. Maybe with this one. I wanted to make them public, but it seems I have them all public already. Okay, never mind. By default, they are not shared, but you can do that. And you can share with your colleagues with this link here. If you just click here, they will disappear from this view, but you are not deleting them from your account. If you want to delete them, you need to click on the trash can. Okay, this is not the only search that you can construct, of course. You can start actually knowing the gene IDs that you want to use. For that, you will go gene ID and start from here and upload a text file. And from there, this is a text file with gene IDs. And from there, start your query and just, add other steps as I show you. What if you do not have the list of gene IDs? You just have characteristics of your genes. So for example, you say, oh, well, I'm interested in genes with transmembrane domains. And I know I want to look in all arthropods. And I'm interested in the ones that have no more than 7 transmembrane domains. And you have it here, and you add another step, and you say, well, and I want them with a specific interpro domain, and I want this in all arthropods, and I want to use interpro scan, and I'm interested in the opsins, so I select RH1, RH2, opsin, RH3 and 4, and I run the steps. So notice I didn't start with a gene ID list. In the second case, I just start with characteristics of it. And you can, keep moving forward with this. In the interest of time, I think I might have to switch because I might have gone over. I'm sorry. Omar, back to you with Galaxy.
Yeah, not a problem. I was actually in the process of typing an answer. So somebody, Gareth Lysett, was asking, how do I combine step 1 and 2 to identify orthologs that are upregulated? And what I was typing, I was just saying that with any search in VectorBase, as Gloria was showing you, you can transform it to orthologs to any other organism in the database. So for example, you can run a search for an RNA sequence search, any of the ones that are available, and ask for genes that are upregulated in one condition compared to the other, and then say, okay, I found my 100 genes that are upregulated in Anopheles, and now you can say, I want to now transform these to orthologs in 80s, right? And so you'll get the orthologs of your upregulated genes in 80s. Now, of course, will they also be upregulated in 80s on the same conditions? You don't know unless you do the real lab experiment, but you can make some good guesses, right? So based on orthology, an ortholog of a gene that's upregulated may also be upregulated under the same conditions. So that's kind of the rationale of using this transform method. Hopefully that answered the question. And I think there are other questions coming in. So keep putting in the questions, and we will answer them online. Okay, so quickly, what I wanted to demo is another feature in the database that allows you to analyze your own data, and then take this, and then of course you can play around with it however you want, but also you can then transfer it back to VectorBase, so you can ask questions about your data in the context of whatever else is in the database. And if you go under this section here, you'll see there's an option for My Workspace. And you can go to Analyze My Data, and this is using the Galaxy platform. So once you click on this, it'll take you to this Galaxy website. If you're familiar with Galaxy, you'll know exactly what this looks like. If you're not, it's actually quite straightforward and not very hard to learn. We do have multiple webinars, as we indicated before, and there is a previous webinar on running a Galaxy workflow in ViewPathDB. So I would recommend that you visit that webinar and learn more. And then there are many resources out there on how to use Galaxy in general, and it's quite powerful. It contains a number of bioinformatic tools, hundreds of bioinformatic tools, and the way you work with these tools is using a web interface. So you don't have to know how to run command line operations. You can do them all using this web interface. And what I'm showing you here on the right is a workflow that I ran yesterday. I just took these 2 samples that Gloria sent to me from a deltamethrin resistant and sensitive Anopheles strains. And I ran them through an RNA sequence pipeline. We have a number of pipelines or workflows available to you here. These are just example workflows that anybody can use, and you can click on those and start using them, or you can work on them and modify them, because, there are different tools out there. Just like in the lab, you may extract DNA using a certain method, and the person in the lab next door may extract the same DNA, but using a slightly different method. It's just their preferred method. And there are many, many different methods out there that you can use. These are just example things to get you started. So I ran this on my analysis, and you get results, here, which you can access. I can go in and start looking at them within the context of Galaxy. But we also have this option to allow you to export data to ViewPathDB. So what I did is and again, I did this morning just to make time so we don't spend time watching this spin. But I exported my RNA sequence data results from my workflow right here. And then what you can do is you go back to, once you export it and it's done, you go back to VectorBase, and under My Workspaces, you'll see here there's an option for My Datasets. And now when I go here, you'll see here I have a couple of data sets that I exported, just testing these around. So this deltamethrin 2 is the one that I'm interested in. So I'm going to click on this. So this is my data set. This is private. Okay, so you have to be logged in. It's under your account, but it's private. Nobody else can see it except for you. And now the cool thing about this is that this is your primary data that you analyzed. And now I can, if I'm interested in knowing, well, show me the genes that are upregulated and resistant compared to sensitive lines, I can click on this RNA-seq user dataset fold change. This gives me a pre-configured query page, which allows me now to configure it and ask, well, I want to find genes that are upregulated. Let's keep it at 2-fold when I compare the sensitive line to the resistant line. So this here on the right is just a visual cue to tell you that a gene that meets this criteria that looks like this will be returned in the results. And then I can run my search. And so what this does, it told me that, oh, there are 141 genes that were upregulated by at least 2-fold in my resistant line compared to my sensitive line. And again, this is based on your own data that you just analyzed in Galaxy. And you'll see here, as Gloria showed you, I'm just going to close this organism filter so we have more space. And you see here that I have the list of my genes. And on the right here, you have a custom graph that gets drawn for you based on your data. So I can see that my, well, depending, I guess I can't remember what parameter I used, but in this case, I'm looking at things that are up and sensitive compared to resistant. Okay, I must have selected it the opposite. And now I can be satisfied and say, okay, I'm done. I have my exciting genes, right? And you're going to work on them and do other things on them. But you can take this a step further and do additional analyses within VectorBase. So one thing I can do is I can simply say, well, where are these genes located? And so if I click on this genome view, it'll show me the chromosomes that have where these genes are coming from, and I can see the distribution of chromosomes. So this is a nice and quick and dirty way for you to see where they are. But also sometimes if there are clusters of genes, this will allow you to see them. I don't see anything obvious here, but sometimes when there's a cluster of genes, you'll see a big blob of blue or red. Each one of these little lines indicates a gene or multiple genes if it's a thicker line. The other thing I can do is I can analyze these results. So I can click on Analyze Results, and you'll notice here that you have an option for different enrichment analyses. If you're interested in learning more about these, we have another webinar that we ran, and it's recorded and available to you here on Go and other enrichment analysis in ViewPath2B. So I'd recommend that you visit that webinar and watch it and learn more about how to do these enrichment analyses. But I can click on Go enrichment, and this will open up a panel here that allows me to configure the Go enrichment analysis. I'm going to select molecular function in this case. I'll keep the p-value at the same and other defaults, and then I submit this. And so this is going to do enrichment analysis. It's looking to see if there's anything, any Go terms enriched in my 141 genes that are upregulated in between the different conditions in comparison to the background, which is the genome. And so now I can go down and look at these, and you'll see here that there's some endopeptidase activity that's upregulated in these lines. Now, whether this is really biologically important or not, you will have to decide. As a scientist, you can take a look at this and ask, well, do these make sense? I have 20 genes in my results that were potentially endopeptidases. Are they involved in delta methrin resistance or not? And as Gloria showed you, you can take this list now and you can add additional steps. So for example, if I'm curious to know, well, out of these 141 genes that are upregulated in my condition, are any of them potentially secreted, right? So I can add a step and just keep the defaults here. I want to intersect my results from my previous step with a new search. And in this case, I'm going to select signal peptide. So I can just start typing signal, and you'll see here you have your predicted signal peptide search. And now I'm just going to select all organisms. The intersect option is selected, and I can run my step. So this will, as this runs now, you'll see that I found over 78,000 genes with a predicted signal peptide in VectorBase, but 62 of them were also found in my list of 141 genes. So I have a good number of potentially secreted genes that are upregulated in my particular condition. And you can add additional steps here. You can compare it to other RNA sequence or microarray experiments in the database, and so forth. I think one of the points to make here before I switch it back to Gloria is that if you're using VectorBase or you're doing something in informatic wise, and you're finding yourself being frustrated by it and you're not knowing what's going on, send us an email, contact us, and ask us if we have a tool that allows you to do this analysis. In many cases we do, and it'll save you a lot of time. In other cases we don't, and it might actually give us some indication of what other tools we need to develop or integrate in the database. All right, so I will end here unless there are any specific questions about Galaxy, and I will switch it back to you, Gloria. Anything else?
Not that I know.
Okay. Perfect.
Okay, show my screen. Okay, you can see my screen again?
Yes. Mm-hmm.
Okay, we have 15 minutes remaining. So I will run through the Genome Browser and the gene pages. So how can you access them? So you can access from the Tools menu, and here you have the Genome Browser, but also from the site search. So you can type a gene ID or a gene name or symbol. So for example, if I type the para gene, and I get here and I say, well, I'm looking actually for a gene that is expressed in Anopheles gambiae. And I click apply, and I see here the top one is the one that I was looking for. And that will load for me the gene page. What do you have? The gene ID at the top. What is the product function? Voltage gated sodium channel. This is the name that I type it, para. It's a protein coding gene. Where is it located? The species, the strain. This is not yet implemented in VectorBase, but this is also a cool new feature for you guys to check later on. You can add there, oh, I have a paper on this gene, or oh, I know the annotation of this gene is wrong, or anything you want to say about a gene. We welcome your comments here. Not yet, but soon. What else I have here? So in this page, you have these shortcuts of quick links of what we think you are the things that you use the most. But also with this navigation here, you can jump to the different sections. So for example, you want to know about this gene has RNA-seq expression or not, and which are the experiments that I have. So I jumped to number 8, and here I have it. And if you have a lot of information on, actually I don't want to see the orthology and synteny, it will just delete it and see how from 6 it jumps to number 8. So this is all the information we have about the genes. And from here, you can link to JBrowse. I'm going to open it here. Oops, sorry. I think I have it open. Yes. So when you arrive here, the people that is already familiar with Apollo can relate to this new genome browser that we will have. Here at the top, you have this navigation bar that will help you to move to one section to another, for example. Here, you can move to the different chromosomes or the scaffolds and supercontigs. And here, actually, notice that we have the chromosome and the base pair range, but you can also just type the name of your gene. If the gene name has been submitted to VectorBase, for example, you are interested in the opsins, and here you have them, and you can just switch to any of them, for example. You can also type the gene ID if you know it. So for example, you want to get AGAP00 something, and the autocomplete will give you all the different options of which the gene that you're looking for. Okay, what else you can do here? So notice we have these magnifying glasses, and the size of them, it's showing you how big of a change you are going to have if you zoom in or out with the different ones. So notice this is the gene right here, but I want to see it with more detail. I actually want to see the exons. Okay. What else we have here? You can move through the windows in 2 different ways. So you can click on the arrows to move left or right. And you can also, with your mouse, just click on the screen and drag. And what I'm doing here is with my trackpad or with your mouse, any way you want to do it. You can select also a feature. For example, you are very interested in this exon right here. So you say, I want to select this. And notice this got yellow. And now this region, so whatever I'm doing down here, I'm always going to know that I'm looking at that region of the there. Something very important is that you can add tracks here. So right now I want to see where the tracks that I have currently active. Okay, so I have RNA-seq data. I'm going to delete all that. I'm just going to delete the annotated transcripts. And I'm going to back. So see how now I only have my genes. Oops, sorry. Here. Only have my genes available. But you want to add things here. So you want to add homology, or for example, I clear all my filters and I have comparative genomics, I have gene models, I have transcriptomics to add, the ones that I had it before. And if you click on any of those, so for each experiment, there are different samples and things that you can add. So you can say, oh, I just want to align the unique ones. Click here and go back to select track. And notice how this RNA-seq evidence really aligns well with the exons that I have predicted. And, could be another case. There is many more things to show about the Genome Browser, but I'm going to stop here. There is, we did a dedicated Genome Browser and Gene Page webinar, and you can go back to where I show you to learn how to use VectorBase and watch it. And now I'm going to quickly tell you about something before I go to Sam that I just telling you.
Hey, Gloria, sorry to interrupt. I think I was just chatting with Sam since we just did a webinar on MapView last week. we thought we'll make the map view portion shorter, so you don't have to rush through this, and so you can take your time. So don't feel like you have to rush.
Okay, I was looking at the clock and say, oh, I have 10 minutes to go.
Take your time, and we'll just refer them to the webinar which we did just last week on map view, and we'll probably be running additional webinars in the future about map view.
Oh, okay, perfect. Okay, so let me show you this. I was just, had it ready. So in the tools menu, you have Apollo, and this just became available yesterday. I know many of you have been emailing the help desk about when can you start annotating your genes, and now it's available. So if you have an account with us for that tool, you do need to have an account to use Apollo, because we need to know who changed the gene and why you did it. For Apollo, we also have a dedicated webinar that Dan Lawson ran like a month ago almost. What do you do in Apollo? So for example, you come here and let's say you realize actually, based on the evidence, it seems like we are missing an exon right here because we have this evidence here, this evidence here, and here that doesn't sound right. Maybe there's some mistake in the annotation, and you want to fix that. So when you want to fix that, that's when you go to Apollo, and there simultaneously everybody can see what you are doing, and we collect all that information and update the gene set. Okay, so back again here. Okay. So now that we have more time, so if I click on one gene, I get more information, and I can actually also download the sequence. So if you just click here, you're not going to get it, but you go here and say, I want the reference. Oops, sorry. There are the filters. The reference. The reference sequence, checkbox, select tracks. Okay, it says zoom in to see sequence. Okay, so I'm going to do that. Select and zoom. And notice I have these different colors, so I'm going to zoom in a little bit more. Okay, so the stop codons are in red and the initiation methionine are in green, all the nucleotides in colors and the different amino acids. And now that you have this track, now you can actually download the sequence for something that you are interested. So for example, that piece of RNA-seq evidence that is puzzling you, that it should be an exon or not. So you you select the sequence. Right again. You select the sequence. Oops. Okay. Not. Sorry. Let me do it again. And sorry, reference sequence. Save the track data. And what do you want? The highlighted region, or you want the visible region? And that region you want to view it. So we'll show you the FASTA sequence that you have highlighted, and you can save that and download that sequence. Okay, what else I was going to show you? Is there anything else, Omar, that I might have forgot that is important about the Genome Browser?
I don't think so. I mean, we do have a whole webinar on the Genome Browser, actually two webinars, one that covers basic Genome JBrowse functionality, and also one that talks about how to explore gene models in the Genome Browser. And both of those are also linked from the webinar page. And I would recommend people to look at there, but we went through sort of details of exactly what you're showing, Gloria. So I think there's enough information there.
Okay, so I think that's all we wanted to show today. I don't know, Omar, are there any questions that need to be answered?
Yeah, so maybe a couple of things. So one is maybe if you could Actually, can I share my screen quickly, and maybe I'll try and answer a question as we do that?
Yes. Let me make you the presenter. Make presenter. Now you are. Oh, sorry. Okay.
There we go. Thank you. So you should see my page. This is the same strategy I was looking at before. There was a question about whether is there a way to see what the sequence length is, and the answer is yes. You can add a column and look at, for example, add CDS length. If you click on add columns, you'll notice here there's really a bewildering number of columns that you can add. It's basically all the data that's available you can add as columns. Imagine this as a really Excel file with hundreds of columns, and you can use this search column option to allow you to find find things that you're interested in. So for example, transcript length or CDS length or protein length, right? So I can add protein length here and transcript length and update columns, and then you will get additional columns that are loaded here corresponding to those. The question though, however, is what if I have hundreds of sequences here and I would like to just take ones of a particular length? And that's actually a little bit more difficult, and we don't have a specific tool that allows you to do that. I would like to, you can obviously download these and just sort them in Excel. That's super easy, obviously, to do. One thing we are working on right now, hopefully we'll develop in the next couple of releases, is an ability to filter on a specific column, right? To say, for example, if you click here and say, only, give me things that are above 1,000 nucleotides and less than 3,000, for example. And that's something that's coming soon. For now, what you would do is you would click on download. And once you click on download, you're offered a number of options. One of these is a tab delimited option. And you can, you can download whatever column you want. So all the columns that you saw before, you can download those. If I clear all, this will just download the IDs. But I can also specifically say I want the IDs plus the length. Right? And now I can select transcript length, protein length, CDS length, and get sequences. And then this will, I'll just do it here. This will open up in a new page, but you can download it obviously as a CSV file, which you can open in Excel. The other cool thing about this download, we didn't talk about this, but I'll mention it here while we're on this page. So remember, I came from my search strategy. I have 62 genes that are upregulated on a particular condition and are potentially secreted. Let's say I'm trying to, I want to investigate the upstream regions of the start sites because I'm interested in looking for maybe regulatory elements that are in common between these 62 genes. I can select FASTA, so this is the sequence file, and now I can say I want to download the genomic sequence, but I can configure this, right? I can say I want to begin at the translation start, and I'm going to go upstream of that translation start, and I'm going to go, let's say, whatever, 500 nucleotides, and I'm going to stop at the translation start as well, because I only want the upstream 500 nucleotides. And now when I get sequences, this is going to, for every single gene in my list, it's only going to give me the upstream 500 nucleotides. And now what I can do is I can take this to another server that does, for example, motif finding. And now I can look for potentially conserved motifs along the in the upstream regions of these genes. So that's a nice, quick way for you to download those sequences. And maybe the last thing, and there may be another question coming in, so obviously we can answer those. And then the last thing I forgot to mention before is that with any of your genes, so as Gloria mentioned, there's a lot of data in the gene pages from all different resources. But if you upload your own data using the Galaxy platform, I showed you that you get this graph when you look at the results. Well, in your transcriptomics section, there will be a new section here for you, which is with your own data. So this is my delta methrin 2 experiment, which I ran, and I can look now at the results of this specific gene and what it looks like. I can open up other transcript data that's available from within the database that we loaded from different experiments. And so in this case, it's an experiment from Sam Rund, who's on the call as well. And so you can this will take a few seconds to load, but you can compare this to your results or look at other experiments, and then your results are loaded here on their own. All right, sorry, Gloria, go ahead.
No, it's okay. I just to tell you that it's 1 minute more, and I don't want to take people's time. So can you make me presenter? Oh, okay, make presenter.
Yes,
we have one more question from Paul. He's asking if Maker is available for identification of new gene models in light of new RNA-seq evidence.
Gloria, do you want to answer or should I answer?
No, go ahead.
So yeah, so Maker, we don't run Maker on our genome as far as I know. We do use, and I would recommend for you to look at the webinar on exploring gene models, and which allows you to leverage RNA sequence data. We do use the RNA sequence data to come up with, well, to map intron spanning reads, and we map them from multiple experiments. So we give you sort of a cumulative result from all the experiments showing you where their intron spanning reads and what the evidence for those intron spanning reads are. So it helps you in defining gene models. And if we, yeah, and that's pretty much it. And that's all available both in the Genome Browser or in Apollo. So if you're interested in doing your own annotation, you can look at those intron spanning reads that we provide evidence for, and that'll help you then and guide you to look for either alternative splice variants of a gene or simply confirming if the gene model is correct.
Omar, so now that we got to the top of the hour, I just have two last things to say. You know, this is what Sam was going to show you again. There is a complete recording about this, but this is just map view. So you can come here and see, you know, insecticide resistance phenotypes, genotype, blood meal, source, pathogen infection status, and many cool things. And Sam did a great job with that webinar last week. So I will strongly encourage you all guys to go and check that. We have a webinar survey. Can somebody from the outreach team please help me post this link in the chat box? We will really appreciate if you could please give us your feedback, because we want to know how we did today, what can we do better, and which new topics you would like for us to cover. So thank you very much to all attendees for your presence and your participation. A final reminder, please remember to cite us in your thesis, your paper, your poster, your talk. This helps us know which of our tools and data sets are more useful to you, and it's also good for our funding agency to know the impact that we have on the community. I don't have anything else to say. I'm going to show the, I think I lost all.
Yeah, Gloria, one quick thing that we will be sending an email tomorrow with you know, a link to the survey as well, and a link to the webinar as well. So you will have an opportunity to do that. For the survey, I'm just putting the link in the chat box, so I'll put it right there and send it to everybody. So you have it. So I would recommend and I would encourage people to do the survey right now. Don't wait. You know, either scan this QR code or click on the link that we just chatted. We know from the last 20 webinars that we've ran, if people do them right away, we get responses. If we don't do it right away, we almost get no responses, and your responses are very valuable to us.
Yes, that's very true, Omar. And thank you also very much for all the outreach members that I didn't mention: Evelina Vashenko at Liverpool University, and you know, Sarah Kelly at the Imperial College of London, and Susanne Warrenfelds at the University of Georgia. They were the ones answering your questions behind the scenes. Everybody have a nice rest of the day, and bye-bye.
Gloria, just a quick question. There's a question about certificate. The certificate will be emailed along with the email after the webinar. So give us a day or two, and you should get it in the mail, in the email.
Great, thank you, Eve.
Bye, everybody.
Thank you. Bye-bye.